Inside every living thing, including you right now, there are trillions of microscopic engines just working away, making life itself possible. These are enzymes, the biological catalysts that literally power our world. So today, let's dive in and see exactly what they do, and maybe more importantly, how they do it. If enzymes could talk, this is pretty much what they'd be screaming from the rooftops. And they'd be right. They are the spark plug for pretty much every single chemical reaction that keeps you alive. Without them, the chemistry of life would just grind to a halt. It would be way too slow to function. Okay, to really get a sense of their power, just look at this comparison. If you want to break down a protein in a lab, you need to boil it in strong acid at 100 degrees Celsius for a couple of days. But inside your body, at a comfy 37 degrees, enzymes get that same job done in just a couple of hours. That's an absolutely mind-blowing jump in efficiency. And that crazy difference really highlights the core problem that enzymes solve. See, most of the chemical reactions that are essential for life, we're talking everything from digestion to copying your DNA, are on their own incredibly slow, way too slow to sustain a living, breathing organism. Without a little push, life would be permanently stuck in the slow lane. So that little push comes from something we call a catalyst. A catalyst is basically any substance that can speed up a reaction, like dramatically, without getting used up in the process. Think of it as a facilitator, a helper that just makes things happen way, way faster than they ever could on their own. Here's a really great way to think about it. A good teacher is a catalyst for learning, right? They don't learn the subject for you, but they guide you, they simplify these super complex ideas, and they help you understand something in a few hours that might have taken you days to figure out on your own. Well, enzymes are doing that exact same thing for the chemical reactions inside your body. And this is where nature's absolutely brilliant solution comes into play: enzymes. They are biological catalysts, and almost all of them are proteins. You can think of them as these tiny, custom-built tools. Each one is folded into a unique 3D shape that perfectly cradles the specific molecules it needs to work on, making it so much easier for that chemical reaction to just happen. Now, because enzymes are such specialists, scientists have actually grouped them into 6 major classes based on the kind of job they do. Some of them transfer chemical groups, others use water to break bonds. Look, the key takeaway here isn't to memorize the names. It's to appreciate the incredible variety in organization. There is a specific tool for pretty much every chemical task you can imagine in the body. And hey, for those of you who are curious or maybe studying for a biochem test, there's even a handy mnemonic to remember these classes: OTHLIL. It's a neat little trick. But for our purposes, what really matters is what these amazing little machines can do. So enzymes are powerful, they're specific, but here's the other amazing thing. They are also incredibly controllable. So let's take a look at their operating manual, if you will. These are the dials and the levers that our biology uses to speed them up or slow them down with just incredible precision. Okay, so the first control dial is what's called substrate concentration. That's basically just how much work you give an enzyme to do. The more stuff you give it to work on, or the more substrate, the faster the reaction goes. But there's a catch. It only goes faster up to a certain point. Eventually, all the enzymes are busy, they're saturated, and the reaction hits its top speed, what scientists call Vmax. Next up, we've got temperature. Every single enzyme has a Goldilocks zone, an optimal temperature where it works best. For us humans, that's right around our normal body temperature. If it gets too cold, they slow right down. That's why we refrigerate food. We're slowing down the bacterial enzymes. But if it gets too hot, they start to fall apart. That's a process called denaturing. And you know what? The exact same principle applies to pH. Every enzyme has its own ideal pH level. So for example, an enzyme that works perfectly in the super high acid environment of your stomach would be totally useless in the neutral pH of your blood. They're all perfectly fine-tuned for their specific little workplace in the body. For centuries, we've really just been observers of this incredible natural machinery. But now we're moving from observation to creation. We are seriously entering an era where we can actually start to engineer enzymes to our own specifications. And this is where it gets really, really exciting. Using genetic engineering, we can now build custom enzymes. We can fuse genes to create hybrid enzymes that make beer brewing more efficient. We can precisely alter an enzyme structure to create a clot-busting drug that lasts longer in the body. We can even create these things called abzymes, which are antibodies that act like enzymes. The possibilities are just exploding. So, we now have the ability to not just harness, but actually improve upon nature's most powerful catalysts. And that leaves us with a huge question. As we get better and better at designing these super enzymes, what problems should we point them at next? Cleaning plastic out of our oceans? Curing genetic diseases? The operating manual is now in our hands. The only question is, what are we going to build?